Hello and welcome back. Today we'll start to discuss chronic inflammation, which is a process of prolonged duration. It may follow acute inflammation or may be more insidious in onset without a preceding acute inflammatory phase, or present between recurrent episodes of acute inflammation. Chronic inflammation is mainly caused by the following conditions: persistent infections by microbes that are difficult to eradicate, such as Mycobacterium tuberculosis, prolonged exposure to endogenous or exogenous toxic agents, for example, the inhalation of silica particles for extended periods causing silicosis, immune-mediated diseases, including autoimmune diseases and allergical diseases, such as rheumatoid arthritis. Chronic inflammation occurs in two major patterns: chronic lung-specific inflammation and granulomatous inflammation. In this session, our topic is chronic lung-specific inflammation. You should be able to recognize the pathological features of chronic lung-specific inflammation. What is an inflammatory polyp and what is an inflammatory pseudotumor? Chronic lung-specific inflammation is morphologically characterized by coexisting ongoing inflammation, tissue injury, and tissue repair, often by fibrosis, to varying degrees. Continuing inflammation reflects the body's persistent response to injury. The principal inflammatory cells are macrophages, lymphocytes, and plasma cells, which are usually called chronic inflammatory cells. Tissue injuries are mainly produced by persistent inflammatory stimuli or products of inflammatory cells. Repair of damaged tissues involves proliferation of vascular epithelium, fibroblasts, epithelium, glands, and other parenchymal cells, usually leading to fibrosis or scarring, which can cause narrowing, obstruction, or sclerosis of organs. Macrophage-lymphocyte interactions in chronic inflammation play an important role in the development and maintenance of chronic inflammation. Macrophages present antigens to lymphocytes and produce cytokines to stimulate T cell responses. Activated T cells, in turn, produce cytokines, such as interferon gamma, to recruit and activate more macrophages, promoting more antigen presentation and cytokine secretion. This results in a cycle of cellular reactions that sustains chronic inflammation. Chronic bronchitis is a typical chronic lung-specific inflammation. These pictures show the microscopic appearance of a chronically inflamed bronchus. We can see bronchial epithelial injury in this area, and some injured cells have come off. The bronchial wall is infiltrated by lymphocytes. For example, this area has dense infiltration of lymphocytes. We can also see glandular hyperplasia. Please note the squamous metaplasia of bronchial epithelium, in which the original ciliated columnar epithelium has been replaced by squamous epithelium. Glandular hyperplasia and squamous metaplasia represent tissue repair. Therefore, in chronic bronchitis, we can identify chronic inflammatory cell infiltration, tissue injury, and tissue repair simultaneously. In this microscopic picture of chronic glomerulonephritis, we can see sclerosis of the glomeruli and marked interstitial fibrosis with infiltration of lymphocytes. In certain sites of the body, chronic lung-specific inflammation may produce characteristic morphologic appearance, such as inflammatory polyp or pseudotumor. Chronic inflammation occurring in the mucosa may form inflammatory polyp. An inflammatory polyp is a mass or lump with a stalk that projects above a mucosal surface. These polyps are formed by excessive inflammatory proliferation of epithelial cells, glands, and granulation tissue. This is a nasal polyp specimen composed of a bunch of gray and translucent nodules strung together. This is a cervical polyp with a stalk adherent to the cervical mucosa. Look at these two pictures. Microscopically, nasal polyp is lined by hyperplastic pseudostratified ciliated columnar epithelium with gland proliferation, massive infiltration of lymphocytes, and mild stromal edema. Here is a microscopic view of a cervical polyp, which contains hyperplastic epithelium, glands, and collective tissue, accompanied by a large amount of lymphocytic infiltrate. There is a prominently dilated gland. An inflammatory pseudotumor is a well-demarcated tumor-like mass formed by excessive proliferation of parenchymal cells and granulation tissue. It often occurs in the orbit and lung. Here we have a tumor-like mass without capsule in the right lower pulmonary lobe. It has been cut open, and what we see is the cut surface of the mass. The cut surface appears yellowish-white mixed with gray-black. This mass encircles part of our bronchus, which has been cut off. The bronchial wall is thickened. Inside the mass, there is a cavity left after loss of necrotic tissue. Clinically, a chest X-ray often reveals pulmonary inflammatory pseudotumor as a solitary, well-circumscribed dense mass. Now, let's make a summary. Chronic inflammation includes chronic lung-specific inflammation and chronic granulomatous inflammation. The latter will be discussed in the next session. Chronic lung-specific inflammation is morphologically characterized by coexisting infiltration of chronic inflammatory cells, tissue injury, and tissue repair, often by fibrosis. In certain sites of the body, chronic lung-specific inflammation may produce characteristic morphological appearance, such as inflammatory polyp or pseudotumor. That's all for now. Here are the references and acknowledgement. Next time, we'll talk about chronic granulomatous inflammation. See you then.